والعلاج وراء كوفيد-19 هو عنوان حديثنا الآن مع الدكتور نبيل صيدا، مدير مختبر الكيمياء الحيوية وعلوم الغدد الصماء معهد مونتريال للبحوث السريرية، لمناقشة العلوم الأساسية وبيولوجيا جائحة كوفيد-19 بالإضافة إلى العلاجات المستقبلية. نبيل حديث 20
شكرا،
ويمكن ترك آخر 5 دقائق للأسئلة.
شكرا جزيلا. أنا النهارده هتكلم بالإنجليزي لأن صعب عشان أحط كل التكنيكال تيرم بالعربي، إنما لو عندكم أي حاجة ما تفهموهاش قوليها لي بالعربي. Today I'm going to be talking about COVID-19, which is a disease that is affecting a worldwide population. How do we understand its biology and what are the available treatments right now? So if one looks at this disease, we realize that these kinds of diseases were very old. In fact, people began to report them about 12,000 years ago, when the human beings began to leave the hunting gathering and began to organize themselves in villages and starting to plant. So a number of plagues and diseases have affected our humanity. As early as the 2nd century, the Antonine plague affected as you can see, the Roman Empire. But some of the plagues have been deadly, really deadly, like the Black Death, bubonic plague, that in the 14th century killed 200 million people. The smallpox have killed 56 million people. The Spanish flu, you may have heard about it. It was in the First World War. It killed around 50 million people. And HIV/AIDS is right now 37 million. So where are we with the SARS coronavirus? We are really a small amount compared to the big pandemics that we knew about. And if you look at in a perspective, the SARS coronavirus has now so far killed 770,000 people. It's still very significant, but it's far away from the huge plagues that affected in history. Some of the plagues are done by specific bacteria, like the Yersinia, which affected the Black Death. Some of them are viruses, like even the Egyptian mummies had these viruses that caused the variola. And some of them are like the flu. So there are different kinds of microbes that will cause these pandemics. The one we are interested in now is called a coronavirus. Why is it called coronavirus? Well, it's called coronavirus because it was discovered early on in 1965. This is an electron microscopy picture, which shows the virus which has crown-like structures. You can see the crown sticks that are around the virus. So it was called a coronavirus. Now, since its discovery in 1965, a number of coronaviruses were also discovered that affect human. 4 of them right now are circulating throughout the world, and they cause a common cold, but they're not lethal. The lethal ones began really to appear in 2002 with the SARS coronavirus, in 2012 with the Middle East Respiratory Syndrome coronavirus, and now the SARS coronavirus number 2. They're all beta coronaviruses and they're highly deadly. Now, if you look at the distribution of the death rate and the infection rate throughout the world right now, it's huge. Almost every country of the world has now experienced infections with this virus. Some of them have dealt with it much better, some of them much less. And you can see right now, as we stand, the United States and South America are the worst hit. Spain is one, Iraq is another one, whereas the other countries like Egypt, Algeria are relatively well treating it. Libya is suffering a lot, and so is India. So if you look at the amount of newly reported death, it's about 23 million people who have been infected by this virus, and about 800,000 deaths so far. But one has to put this into perspective. If one looks at the normal death rate around the year in every country, you can see that in some countries, even though they were infected by the coronavirus, it didn't really affect the average weekly death in the population. But countries like Peru, Chile, South Africa, Jakarta, Ecuador, Italy, they did experience a peak where there was much more death than would have been expected from the normal death. And if one calculates how much is the difference, you see that the United States is the one that has the highest difference between the normal amount of death and the actual people who die. But if you look at it in terms of the population, it's about only 20%. Peru and Mexico City are really much more affected. So one has to get a relative view of this virus. It is a very deadly virus, and we are really trying to contain it. How does it get transmitted? Well, this virus is not transmitted like by a mosquito, like the case of the flu virus. It is rather transmitted by coughing or by droplets after you basically sneeze. It's called the fomites. And you get small and large droplets. The small droplets go further away than the large droplets, and they can then affect the respiratory system of the individuals. If they are on hands or on beds, you can get direct contacts with the human. Now you can see the virus inside the droplet. Here is a figure, and it actually is transmitted this way as you breathe. Now, if one looks at the amount of death caused by this virus, we estimate around 4% of the individuals who were infected by the virus so far actually died. But this may be an overestimate because we haven't tested everybody which do not show symptoms. In Italy and in other countries, we noticed that this virus is different from the flu virus. It's really affecting the older population. And 99% of the people who die from this virus are around 60 to 90 years of age. All right, so what they tell us, everybody, is to be able to avoid infection of this virus, wash your hands and wear a mask. Now, you wash your hands because the soap breaks down the barriers of the virus. You can see it. And the mask prevents the virus from spreading and at least transmitting from human to human. Now, what is a coronavirus? What is that coronavirus? Can we understand its biology and maybe develop much more efficient treatment? As you know, in our world, we are a world of DNA that is transformed into ribonucleic acid, RNA, and then that is coded into proteins. Well, this virus is an RNA virus, okay? It's already doesn't need DNA. It's an RNA virus, a single-strand RNA virus. Its length is not so big. It's around 30,000 nucleotides, and it encodes 26 proteins. 26 proteins are affecting the whole world. It's amazing, okay? So when one looks at the subgenomic structure of this virus, you can see the virus that you see in the electron microscope. You can see the spikes, what we call the S or the spike, is what you see in the corona part. But really the 26 proteins, they do different things. And people who are trying to develop treatments against this virus are trying to attack different parts of the virus. They're trying to attack the protease of the virus, the RNA-dependent RNA polymerase, the helicase, the nuclease. And we in our lab are really interested in the S or the spike glycoprotein, which allows the virus to enter the lungs. Now, we discovered very early on that this was a special virus because when you looked at the sequence of the spike glycoprotein, we noticed that there was a 4 amino acid insertion. That made all the difference why this virus is so infectious. By the way, the same thing was observed in the MERS, the Middle East Respiratory Syndrome, except there, the virus is not jumping from human to human, but it's remaining from camel to human. Whereas in the SARS coronavirus, it goes from human to human. So, of course, in order to develop effective therapy against this virus, you have to understand how does it get in? How does it infect? So we are interested in the way the viral enters the cells and how does it cause infectivity to every other tissue. So in order for a virus infection to happen, you need the virus to bind a host cell. In this case, is the lungs, for example, or the nose, typically by interaction with cellular receptors. In this case, it's going to be angiotensin converting enzyme 2 called ACE2. Then once it's interacted with the receptor, it will fuse and deliver the cargo inside the cell. And once it delivers the cargo, it can then multiply itself, come out, and infect other cells. So if you look at it the way we in the labs are looking at the virus, and here comes the virus, it looks for a receptor. It's basically like a key and a lock. The lock would be the ACE2, and the key is the virus. Once the key enters the lock, it can then enter the cell. So it turns out this virus is really difficult to handle because it uses two ways to get in. Once is used the endocytosis way, it goes directly in. And the other way, it uses what is known as diffusion. It has to be cleaved. The S-glycoprotein has to be cleaved by two enzymes, furin and TMPRSS2, for it to fuse. Once it is fused, it will then deliver the cargo, which will start making new viruses. So one of the approaches to block the virus has been to block its entry. And people, as well as us, have been working very hard to block the entry of the virus. And we discovered that you have to block the two enzymes, furin and TMPRSS2. If you can block the two of them, the virus has very difficult time getting into the lung. So this is probably a future antiviral. Now, you worry because you say, oh my God, if one looks at what happens in the HIV virus and other viruses, every time you try to develop a drug, the virus mutates. It changes its structure so that the drug is not effective after 6 months. So it turns out that this virus, and luckily for all of us, okay, has what is known as a proofreader. In other words, it's like when you copy a text, you make sure that there are no errors. So it has a proofreading capacity that makes only 2 mutations on 30,000 every month. So that means you have a very low rate of mutation. This is good news because it means that our treatments will probably last. But it did find mutations, and there is one mutation particularly is a glutamine at position 614, an aspartic at position 614 that is replaced by glycine. It first started when it came from China originally, it had the aspartic. And now all over the world, it is the glycine derivative that is now making. So even though it is a low fusogenic and mutational functions, it does mutate. And when it does mutate, apparently this mutation makes the virus much more infectious. So we have to be aware that the viruses will try to get more infections with time. Now, how do we go around this? So how does our body react when you have an infection like that? We have what is known as immunity. Our immune system will try to stop this viral infection. And there are 2 types of immunity. An innate immunity, which is a cellular immunity that happens within minutes after you're infected, trying to stall the viral functions. And then there is another one that happens weeks later, 2 to 3 weeks later, to develop antibodies. It's called an adaptive immunity. So this is what the treatments are all about. The innate one, the early immunity, is what causes what is known as the cytokine storm. It's what kills. What people die is not from the virus, but from the overreaction to the virus. Okay, our system will overreact by the innate immunity, called what is known as the cytokine storm. And that will cause cardiovascular complication because it turns out that you have much less oxygen, you have the difficulty in breathing, you have a blockage of the blood vessels, and you get a coagulation, blood coagulation. And so the cytokine storm, for you to understand, is an overreaction to the viral. It's a viral response phase. And once you get the cytokine storm, you affect a lot of tissues. You affect the kidneys, you affect the nose, you affect the circulatory functions, you have fever, you have fatigues. It all has to do with our ability to maintain the virus in check, but sometimes and very often we do overreact. So when you look at patients that come to the clinic, you have 3 situations where the doctor has to decide how to treat them. And so there are a number of clinical trials right now being tried to block the infectivity. First of all, if you have a very acute situation where you have problems of breathing and so on, you have hypoxemia, you require ventilators, you require blood coagulation thinners, and it may progress to a very serious disease and you die. Other patients can be asymptomatic. The younger children and so on are asymptomatic. They have the virus, but they're able to maintain it because their innate immune system doesn't overreact, okay? And they reproduce antibodies. These people can get lower respiratory tract infection and they may require ventilators. But if you're going to make a vaccine, you're going to have to give it to healthy volunteers. You have to give it to the general population. Because once the virus is in, your vaccine is not functional anymore. Okay, you have to do it before. It's a protective mechanism. And those that are going to be mostly targeted are anybody who's above 60, obese, hypertensive, type 2 diabetes, asthmatic, or other cardiovascular complications such as myocardial infarct or cancer patients. So these are the ones you want to target first because they have much more sensitivity to the virus. Now, this is what is the landscape of everybody else in the world who is trying to develop techniques to block this virus. So you can block it from entry, as I was telling you, with the furin inhibitors, or now in Russia, it was discovered 25 years ago, a compound called arbidol that prevents the fusion of the virus with the cells. You can attack the non-structural protein number 13, which is called the helicase, which allows the RNA to unwind, and you can block the ability of the virus to multiply. You can block the enzymes that are responsible for translating the RNA into a protein and cleaving the proteins. And also you can block into the RNA-dependent RNA polymerase for translation. So all these steps, every drug that's being developed around is targeting one or more of these steps. But because of the people that get cytokine storm, people try to prevent the agents implicated in cytokine storm. These are called the cytokines. So they now have interleukin-6 receptor inhibitors, TNF-alpha receptor inhibitors. These are also available. But I have to tell you that none of these treatments eradicated the virus. They are all prophylactic, if you want, so far. So people are putting a lot of hope on vaccine right now, okay? But a vaccine, as you know, is composed of two components. There is the antigen, that in this case is going to be the S protein, the surface protein, the spike. And you have to put an adjuvant that will make the immune system respond much faster. This is how you make a vaccine. So you can make a vaccine, and I'll tell you about it. And you can also make artificially antibodies in the lab. You don't have to wait people to get vaccinated. You can actually produce antibodies that can be injected to humans. This is called the neutralizing monoclonal antibodies. So as we speak, there's more than 140 vaccines that are being tested around the world. 5 are in phase 3. That means that there is 30,000 people being injected or more. So far, 0 are approved. We don't know yet the consequences of these vaccines. So there are different ways to develop a vaccine. You can do that by the inactivated virus, and that's what's mostly happening in China. They have injected the inactivated virus, the old ways, how we used to make vaccines. You can use viral vectors that will transport certain part of the protein. You can use DNA vaccines. You can produce the particles in plants or in other ways in virus-like particles, and you can actually inject the protein. All these approaches will force our immune system to develop antibodies, and hopefully some of these antibodies are going to be neutralizing. So this is the latest story of what is happening. So you can see that there is 141 vaccine in development, 25 are in human trials, only 5 so far are in what we call phase 3. These are the serious ones. There are 30,000 people or more. So in China, they're dominating in a way. They're using inactivated virus. You've heard about the Moderna in Boston. They're using an mRNA virus. And the Oxford and AstraZeneca in England, they're using an adenovirus delivery of the S-glycoprotein. Other people are using different approaches. Merck is using a vesicular stomatitis virus approach. Even people perfuse proteins like Novavax. So we have all these trials. Which one is going to succeed? We'll have to wait. So far, the most advanced is the Moderna, which apparently will begin to give us data soon. The University of Oxford is going to be soon also. The Pfizer is going to be in September. Okay. And then there is this other viruses, and I don't think really we will get a really clear picture of what is going on before 2021. Okay. So I think it's a lot of wishful thinking to try to push it earlier than that, but I think it's going to take the time. The recombinant approach doesn't have to wait for a vaccine. So the recombinant, what you do is you produce the antibody in the lab and you actually produce it in large amounts that you can inject. So there are two companies that are really advanced in this. Regeneron did a nice trick. They combine two antibodies together to hit two parts of the protein. And that's way they will not, even if the virus mutates, it cannot mutate at the two parts at the same time. And Eli Lilly, what they did, they took a patient that was infected, isolated the antibodies, and made a drug out of it, a monoclonal antibody. So these are the two that are advanced. We'll see what's going to happen. The antivirals, as I showed you in the presentation, are now we hear in the news about remdesivir. Remdesivir basically is by Gilead, a company that made its RNA-dependent RNA polymerase, the non-structural protein number 12. It's working, but it's only diminishing by 4 days the time you spend in the clinic, but it doesn't diminish death. It doesn't actually prevent death. Now they're combining it with interferon 3, which is an antiviral. We will see what's going to happen. The viral proteases are still very early on. It's not, we do not know. There are people trying to hit them, as well as the helicase. In my group and in others in England and in Germany, we are using anti-host protease inhibitors, the TMPRSS2, furin, and the catepsis. Other people say, why? What about hitting the receptor? What about preventing the virus from getting into the receptor? And this is where the Russians have this arbidol, which prevent fusion. People said, okay, you got infected. So what do? You get a lot of inflammation. Maybe we can diminish the amount of inflammation, this overreaction, by putting anti-inflammatory agents. In fact, there is tocilizumab, which are interleukin-6 receptor antagonists, if you want, or interleukin-1 receptor. You hear about in the news dexamethasone. This one was good. It's a glucocorticoid. It diminishes by 25% the incidence of death in people infected. Colchicine is another one that people use for myocardial infarct patients. And even the anti-inflammatory, which is broad activity, such as naproxen. Now, we are saying that because you are infected and you get this inflammation, you get a lot of oxygen, free oxygen. So you need to remove this oxygen. So people are using antioxidants, such as glutathione, N-acetylcysteine, superoxide dismutase combinations. Other people think that we can actually use vitamins. And in fact, it is suggested that people who are working with patients who have COVID in emergency department or intensive care units are suggested to take vitamin C and D and zinc, as well as maybe hydroxychloroquine that you hear about. Finally, there is one that is very much in development, which is an infusion of stem cells, which prevent lung damage. Then there is the decoy protein, and the latest one is to actually silence the virus in cells which are infected. So let me just give you what I view as what is happening. So the world continues struggles against the COVID-19. More than 23 million cases and close to 800,000 deaths today have been reported worldwide. When will it end? I must say that rarely we have seen in a situation where basic knowledge, we're just learning everything about this virus. It's all new. It doesn't resemble the other viruses. So the knowledge has to be acquired at the same time as we are developing techniques to eradicate it. So it's a trial and error, if you want. But there are positives that came out. First of all, COVID-19 is beginning to be under control in many countries. Second thing, it has changed our way of life, temporarily at least. We have all abandoned our ways where we used to be in large groups. Unfortunately, we have to distance ourselves, but this is the way to prevent transmission. We have learned how to manage this disease. We have learned how to diminish the coagulation, and novel effective treatments are hopefully expected in the near future. We have learned how to act in unison across the world. Whether you go to Egypt, you go to any other country, we all seem to act in similar ways to prevent this global scale direction. And now, finally, we also know our different difficulties that we have, how our body reacts, and we're beginning to manage them. So stay safe, and thank you for listening. So I see that there are some questions where they say I have to mix Arabic and English. You can see how difficult the subjects are. There are no good translation words for every parts that I presented, but لو عندكم حاجات ما تفهمهاش ممكن أرد عليها بالعربي. So one of the questions was أي فاكسين هيبقى أحسن واحد؟ صعب علينا إن نعرف اللي هيحصل إيه. بيكون مرات secondary effects ما بنعرفهاش لسه. فلازم نستنى شوية نشوف. أنا probably December, January هنعرف هيحصل إيه. Russia have injected attenuated virus to the army, and they have bypassed completely the clinical trials. This is very dangerous precedent. We don't know what's going to happen. To be honest, this is not the way to do science. This is the way to try to pass a virus without knowing its consequences. Now, one of the questions was, why is it that some people have a much stronger reaction or cytokine storm and others don't? It's a very complex question, very difficult to answer. It has to do with our genome, what we inherited from our father and mother. It also has to do with maybe our environment, okay? If our environment is not healthy, we may have more than one pathogens that affect us. But it's still why is it that children can control much better this virus than us? We think it has to do with their innate immune response, which is much more robust than ours, and it doesn't overreact. And if we could control our innate immune response, and there are a number of people trying to do this, okay, maybe we should be able to do it. The question is, why not viruses infect babies? It's not true. Viruses do infect babies, but because babies have the angiotensin converting enzyme 2 substrate receptor, but it's much a lower level. They have a much lower level of ACE2. So that means the infection is much lower and their immune system is much better. Why is the viral increasing in the USA and other advanced countries? Well, it has to do with containment. It has to do with the way we distance ourselves. It has to do with the way we have to respect what science is telling us and not try to think that science doesn't know what it's doing. In the United States and other countries, as well as England, they waited too long. They should have caused the distancing. Look what happened in Japan. Look what happened in Korea. They understood very early on because they've seen viruses before. Look what happened in Africa. They have a lot of pandemics in Africa. they do the best to do that as soon as possible. The United States, unfortunately, did not react on time, and they're paying the price right now. So will we get a vaccine before Russia? I don't know. Will China be ahead? We don't know. We still don't know all the consequences of this virus also. You know, there are all types of problems with the vaccine. It's not necessarily that it will work. And also, how long would the vaccine last? So there are so many unanswered questions that unfortunately we're going to have to wait. Time will tell. I would love to have the answer for you, but nobody has. See, the Russians bypassed completely the clinical trials and they went straight to the patient. We shall see. This is not the way to do science. This is very dangerous. You're going to get all types of complications that maybe we'll hear about it. Yeah, so the question is, can we get comorbidities? In other words, can we get infected by the coronavirus and another virus at the same time? I was just telling you that people who have, for example, HIV infection and that they will have coronavirus, this can be a comorbidity. It can make things much worse. Or influenza. Interestingly, in this year, the amount of influenza, people dying from influenza has been less than years before, maybe because it's competing with this virus. Yeah, so why is China able to contain this epidemic so quickly? To be honest, I'm not so sure that they're telling us the whole story, okay? I'm not so sure that we have all the data in a very transparent way, because it's very strange that they went to up to 80,000 and all at once it stopped. I think there is lack of transparency here. We hope that it's true, okay? But I believe there is a lack of transparency. So I think we should take this with a grain of salt. What can we do in our countries, in the Arab countries and so on, to avoid infection? Well, clearly, to avoid infection, you need to wash your hands very often, like 5 to 10 times a day. You, unfortunately, and believe me, nobody likes it, have to wear your mask, okay? And try not to gather in large groups, because some person in that group may have no symptoms. It could be an asymptomatic, but still transmit the disease. In other words, its immune system, its innate immune system is not overreacting, but the virus is still there. And if it coughs or it talks with high loud language, you may get infected. So please protect yourself and protect your children and your grandparents, because they are the ones that are more sensitive to this virus.